right, so now we're going to move into our chapter 4. And this is all about prokaryotic diversity, as it is entitled. So what this chapter is basically focusing on is identifying and classifying prokaryotic organisms. So we're going to go through this, and I think that what would be helpful is for you to use some flashcards and maybe write the name of the type on the front, and then some characteristics of it on the back, and then just take some time going through these, because it does take a little bit of time. I'm going to go through them, but it's a lot of information. It's a lot of identifying and classifying. So our introduction is just saying scientists have studied prokaryotes for centuries, but it wasn't until 1966 that Thomas Brock discovered that certain bacteria can live in boiling water. Excuse me. So this led many to wonder whether prokaryotes may be able to live in other extreme environments, such as the bottom of the ocean, high altitudes, inside volcanoes, or even on other planets. We have gone through and done a lot of research since 1966 to find out that there are, in fact, lots of prokaryotic organisms in those areas. So prokaryotes have an important role in changing, shaping, and sustaining the entire biosphere. They can produce proteins and other substances used by molecular biologists in basic research and in medicine and industry. So we see a lot of that being used in medicine and industry, like we've already talked about with pasture and other things like penicillin. We found that that is very helpful. And also here it says, for example, the bacterium Shewanella lives in the deep sea. So we have found some in the deep sea again, where oxygen is scarce. So it grows long appendages, which have special sensors that are used to seek out the limited oxygen in its environment. So it has adapted to living in the deep sea. So it has these long appendages that are going to sense oxygen in its surroundings and then utilize that oxygen for it to be able to function and metabolize and live. It can also digest toxic waste and generate electricity. So other species of prokaryotes can produce more oxygen than the entire Amazon rainforest. So there are others like cyanobacteria that produce mass quantities of oxygen that are very, very helpful for our environment. While still others supply plants, animals, and humans with usable forms of nitrogen. So those that can produce nitrogen are very helpful. They inhabit our body, protecting us from harmful microorganisms. So we have microorganisms in our body, in our intestines, in our stomach, in our mouths, that protect us from other microorganisms. And they also produce some vitally important substances. So for example, there's a bacterium in our intestine. It helps us produce vitamin K, which is important. So this chapter will examine the diversity, the structure, and the function of prokaryotes. All right, so first, prokaryote habitats, relationships, and microbiomes. So here's some general information that then will be applied to a lot of the microorganisms, a lot of the prokaryotes in this chapter. Again, all living organisms are classified into the 3 domains of life: archaea, bacteria, and eukarya. And then in this chapter, we're going to focus on archaea and bacteria. They are unicellular prokaryotic organisms. Unlike eukaryotes, they have no nuclei and no other membrane-bound organelles. All right, you should already know that. So prokaryote habitats and functions. Prokaryotes are ubiquitous. They can be found everywhere on our planet. So even hot springs in the Antarctic ice shield, under extreme pressure that are 2 miles underneath water. So we find bacteria, or not bacteria, but we find prokaryotes all over the planet, everywhere. One bacterium, for example, Paracoccus denitrificans, has even been shown to survive when scientists remove it from its native environment, which was in the soil, used a centrifuge to subject it to forces of gravity as strong as those found on the surface of Jupiter. So even though this bacteria isn't specifically designed to live in an area that has this force of gravity, the scientists, when they removed it and subjected it to that type of gravity, they still were able to survive. So prokaryotes are very hardy. Lots of them are very hardy and can live in some extreme environments, some extreme locations. Prokaryotes are also abundant on and within the human body. According to a report by the National Institutes of Health, prokaryotes, especially bacteria, outnumber human cells 10 to 1. So that's something that has been said for many years, is that there are 10 times more prokaryotic cells in and on the human body than there are human cells. And then, however, this says most recent studies suggest that the ratio may be closer to a 1 to 1 rather than a 10 to 1. But as they mention here, even that ratio means there are an incredible amount of bacteria living in and on our body right now. Bacteria thrive in the human mouth, for example, the nasal cavity, the throat, in the ears, the GI tract, gastrointestinal tract, which is the digestive tract, and the vagina. Large colonies of bacteria can be found on healthy human skin. So even on our human skin, even though we wash our hands and clean our bodies, on the human skin we have bacteria that are living there, especially in moist areas that get a little bit smelly, right? Armpits, navel, behind the ears. However, even drier areas of the skin are not free from bacteria. So even just the arm, on the arm or on the leg or ankle, those kinds of things that don't get smelly when we haven't showered for a while or go through some sort of heavy activity. So the existence of prokaryotes is very important for the stability and the thriving of ecosystems. So they play a key role in all of this web of life on this planet. As we mentioned already, our human body would not function the same way if we did not have prokaryotes in it and on it. Also, as was mentioned briefly a moment ago, things like cyanobacteria and other bacteria that produce oxygen, our environment would not be what it is today if we didn't have those. So it's very important that we have prokaryotes in our environment on the planet because that is how a lot of the things function. That's how they're the backbone, oftentimes, of many of the things in this earth. So, for example, they show here, they are a necessary part of soil formation. So that's one I didn't use yet. So soil formation and stabilization process through the breakdown of organic matter. So breaking down that organic matter in the soil is very important, and development of biofilms. So that is also a very important process. In order for plants, for example, to get the nutrients from the soil, they need to be broken down sometimes, and bacteria are there to do that. So 1 gram of soil contains up to 10 billion microorganisms. So if you can just picture a small amount of soil has 10 billion microorganisms, most of them prokaryotic, belonging to about 1,000 species. Many species of bacteria use substances released from plant roots, such as acids and carbohydrates, as nutrients. So a lot of the reason that we see the bacteria in there is because they use what the plants are providing. So their nutrients they're getting from the plant's roots, such as these acids and carbohydrates, and then that's what they metabolize in order to live. So the bacteria metabolize these plant substances, and then they release the products of bacterial metabolism back into the soil, and then that can form humus and increase the soil's fertility. So this process, the plant is helping the bacteria by providing these nutrients, by providing the acids, providing the carbohydrates. The bacteria are able to metabolize those substances and then release their metabolized products back into the soil, and increasing the fertility of the soil, which then is going to allow the plants to produce seed and then produce more plants. In salty lakes, such as the Dead Sea, we have our salt-loving halobacteria. They decompose dead brine shrimp. So as the brine shrimp are dying, they decompose them, break them down, and then that nourishes young brine shrimp and the flies with the products of the bacterial metabolism. So in the same way we see, not with plants, but in this case with the brine shrimp. So in these very salty, salt-loving sea, the Dead Sea, we see these bacteria, the halobacteria, that are going to use the dead brine shrimp, break them down, use them to metabolize for their own function so that they can stay alive, and then what they produce, their waste, as it were, is going to go to nourish young brine shrimp. So another relationship there. So in addition to living in the ground and in the water, prokaryotic microorganisms are abundant in the air, even high in the atmosphere. So there may be up to 2,000 different kinds of bacteria in the air, similar to the diversity in the soil. So we see them flying around in the air. Well, we don't see them with our eyes, but we know that they are in the air. It's been tested through growing them on an agar plate, for example, and then looking at them through a microscope. And there are many, many different kinds, so up to maybe 2,000 different kinds floating around. Prokaryotes can be found everywhere on Earth because they are extremely resilient and adaptable. So as we saw with the one that could live in forces of gravity, like on Jupiter, it wasn't living in that type of atmosphere. It hasn't because we don't have that type of gravity here. But because it is adaptable and very resilient, it was able to survive in that kind of atmosphere, in that kind of gravity. They are often metabolically flexible, which means they might easily switch from one energy source to another, depending on the availability of the sources, or from one metabolic pathway to another. So a lot of times microorganisms, a lot of times prokaryotes, bacteria, are able to utilize multiple things for metabolism. So once they've used up their preferred form, then they can switch and use something else to be able to continue to grow and live. Otherwise, they also can switch from one metabolic pathway to another. So if they have the particular source, the energy source, and they go through a preferred pathway of being able to metabolize that, but then that pathway depends on something else that isn't available, then they can switch to a different metabolic pathway using that same energy source, but metabolizing it, breaking it down in a different way. For example, certain prokaryotic cyanobacteria can switch from a conventional type of lipid metabolism, so fat metabolism, which includes production of fatty aldehydes, to a different type of lipid metabolism that generates biofuel, such as fatty acids and wax esters. So in this case, it's going to switch. It's going to go from one type of lipid metabolism that produces these fatty aldehydes, and then go to a different type of lipid metabolism that generates biofuel, which are fatty acids and wax esters. Groundwater bacteria store complex high-energy carbohydrates when grown in pure groundwater, but they metabolize these molecules when the groundwater is enriched with phosphates. So again, it's switching. Some bacteria get their energy by reducing sulfates into sulfides, but can then switch to a different metabolic pathway when necessary, producing acids and free hydrogen ions. So prokaryotes perform functions vital to life on Earth by capturing, or what is called fixing, and recycling elements like carbon and nitrogen. So we'll see a lot of nitrogen-fixing bacteria or carbon-fixing bacteria. And that means that they are going to capture those things, capture the carbon or capture the nitrogen, and then they're going to recycle them. They're going to go through a metabolic process, and then through that metabolic process, it can then produce other types of carbon or nitrogen. So organisms such as animals require organic carbon to grow, but unlike prokaryotes, they are unable to use inorganic carbon sources, like carbon dioxide. Thus, animals rely on prokaryotes to convert the carbon dioxide, which remember is the inorganic carbon, so carbon dioxide, which us humans are unable to use, into organic carbon products that us as humans and other animals can use. So this process of converting carbon dioxide to organic carbon products is called carbon fixation. So in this example here, they're saying that humans, we are unable to use carbon dioxide as a form of building block in our body. We're not able to separate the carbon and the oxygen molecules in order to use that carbon and build with it. So we rely on prokaryotes to take that carbon dioxide out of the air, break it down, and then get rid of it as their waste in a different form, in an organic carbon form that then we can use. We can take in the organic carbon and then we can use that as our building block. So plants and animals also rely heavily on prokaryotes for nitrogen fixation. So we have carbon fixation, and now we're talking about nitrogen fixation. So this is the conversion of atmospheric nitrogen into ammonia. a compound that some plants can use to form many different biomolecules that are necessary for their survival. So, in this case, the bacteria, or the prokaryote, is going to be taking nitrogen that's found in the atmosphere. Air is partially nitrogen, so they can take the nitrogen from the atmosphere, from the air, and convert it into ammonia, and then that's useful for some plants. So bacteria in the genus Rhizobium, for example, are nitrogen-fixing bacteria, and they live in the roots of legume plants. So they actually have these little nodules, and they live in these little nodules, these Rhizobium bacteria. They live in clover, alfalfa, and peas, and then the ammonia that's produced by these Rhizobium help these plants survive by enabling them to make building blocks of nucleic acids. So, and actually I think there's a picture down here. Yeah, you can see the Rhizobium right here in A. You can see these little root nodules, and those are little outgrowths of the roots, and within there, they're like little packets of lots of the Rhizobium bacteria. They live in these root nodules of these legumes. And then over here in B, you can see this micrograph of the root nodule that shows the bacteroids, which are bacterium-like cells, or the modified bacterial cells, that are going to be producing the nitrogen that the plant can then use. So, again, the bacteria Rhizobium is going to live within those roots. They have this relationship where they are going to take nitrogen from the atmosphere, and then they produce ammonia, and then the ammonia is useful to the plant because then the plant can take the nitrogen from it and make nucleic acids. So in turn, these plants may be eaten by animals, sustaining their growth and survival, or they may die, in which case the products of nitrogen fixation will enrich the soil and be used by other plants. So let's pretend it's alfalfa or something, and then the human or the other animal is going to eat that alfalfa, so we have this nice plant growing. Eat the alfalfa, and then it's going to give energy to that animal. Or if it is not eaten by another animal, it may die, and then it just gets recycled in the soil, which is going to help enrich the soil and make it healthier for other plants and other bacteria. Another positive function of prokaryotes is in cleaning up the environment. So recently, some researchers have focused on the diversity and function of prokaryotes in man-made environments. So they found that some bacteria play a unique role in degrading toxic chemicals that pollute water and soil. So a lot of researchers are looking at using different types of bacteria to clean up the damage we've done in the water and the soil. So for example, they're looking at bacteria to help clean up oil slicks. So that's one way that we're looking at prokaryotes to kind of play a role. Despite all of the positive and helpful roles prokaryotes play, some, of course, are human pathogens that may cause illness or infection when they enter the body. So some bacteria also can contaminate food, causing spoilage or foodborne illness, which makes them the subject of concern in food preparation. Less than 1% of prokaryotes, and all of them are bacteria, are thought to be human pathogens. So even though we hear a lot about bacteria and infections and making sure we wash our hands and don't cough on someone else and be careful of blood and all of these different things, really only 1% or less are actual human pathogens. But this 1% are responsible for a large number of the diseases that afflict humans. So the other 99% plus are either helpful or just not helpful or harmful. Besides pathogens, which have a direct impact on human health, prokaryotes also affect humans in indirect ways. So, for example, prokaryotes are now thought to be key players in the process of climate change. So they're really looking into the role that prokaryotes have in climate change. So what they found is recently, as these temperatures in the Earth's polar regions, the poles, have risen, soil that was formerly frozen year-round, which is called permafrost, so the soil that never ever softened and warmed up, has always been frozen, has started to thaw because of these rising temperatures. What we have been seeing then is that carbon trapped in this permafrost, this frozen soil, is gradually released and metabolized by prokaryotes. So as this carbon that's been trapped and frozen for many years is released, it's metabolized by prokaryotes. And then this produces massive amounts of carbon dioxide and methane, which are the greenhouse gases that are going into the atmosphere and then contributing to the greenhouse effect. So as temperatures are rising, we see this melt. The melt is then providing food for lots of prokaryotes that then are producing this carbon dioxide and methane. So that's something that's being looked into as well. So we see a lot of help and a lot of great things for humans and animals in the earth from prokaryotic organisms, but then also we have some other things that we look into. So less than 1% that can actually harm a human, for example, and then looking at some environmental issues. All right, so let's take a look at some of the relationships. We've talked about a lot of the relationships here, but what we're going to do is classify those relationships. So first, we're going to take a look at the different types of relationships and what that means. So we have learned that prokaryotic microorganisms can associate with plants and animals, and then this creates this unique relationship between these organisms. For example, the bacteria living on the roots or leaves of the plant gets nutrients from the plant and in turn produces substances that protect the plant from pathogens. On the other hand, some bacteria are plant pathogens, and they use mechanisms of infection to then harm the plant, just like we see in animals and in humans. So prokaryotes live in what's called a community, which is a group of interacting populations of organisms. So there are so many prokaryotes living on the planet, and they end up living in these communities where there's a mix of them or a lot of one type in a certain area. So a group of interacting populations of organisms. A population is a group of individual organisms belonging to the same biological species and limited to a certain geographic area. So we have a population, which is a group of all the same type of biological species in a certain area that is interacting with other populations. So populations can have cooperative interactions, which benefit the populations, or they can have competitive interactions in which the populations are going to be competing. So one population competes with the other one for resources. So those are two larger, more general categories here. Cooperative interaction, where they benefit both populations, or competitive, where they compete. So the study of these interactions between populations is called microbial ecology. So ecology in the environment, and microbial. So we're talking about environmental microbial interactions. Any interaction between different species within a community is called symbiosis. Okay, so we're talking about our symbioses here. Such interactions fall along a continuum between opposition and cooperation. So interactions in a symbiotic relationship may be beneficial or harmful or have no effect on one or both of the species involved. So this table right here kind of sums up what the next couple of paragraphs we'll be talking about. So here's a certain type of symbiotic relationship right here, the type column, and then this is how it is, the interaction between one population and the other population. So very general interactions here. So for example, the first one we'll look at is mutualism, and in mutualism, population A is benefited and so is population B. So both are benefited in a mutualistic symbiosis. And then we're going to go through each one of these here in a little bit more detail. But that's a nice little summarization. So when two species benefit from each other, that's called mutualism, as we just said. So for example, humans have a mutualistic relationship with the bacterium, this guy here, Bacteroides theta iota tetra iota micron, which lives in the intestinal tract. So this guy digests complex polysaccharide plant materials that human digestive enzymes cannot break down. And then it converts them into monosaccharides that can be absorbed by human cells. So things like cellulose. When we eat plant material, when humans eat plant material, our actual body cells are unable to break down the cellulose walls in plant material. But we have this bacterium that hangs out in our digestive system, and it eats up the plant material, the cellulose. It can break those bonds, and it converts them into monosaccharides, which is a single sugar that we can absorb through our human cells. And so that is incredibly helpful. Otherwise, we would be eating plants after plants after plants after plant and not be getting the benefit from that. Humans also have a mutualistic relationship with certain strains of E. coli, Escherichia coli, another bacterium found in the gut. So E. coli relies on intestinal contents for nutrients, and then the humans derive certain vitamins from E. coli, so vitamin K, which I had already mentioned. So this is required for the formation of blood clotting factors. And then it does note here, of course, we don't think of E. coli as something that we generally want in our bodies. Perhaps you're used to hearing bad things about E. coli. And that's true. This is only true for some strains of E. coli. However, other strains are pathogenic, and we don't have a mutualistic relationship with those because, remember, mutualism is based on both populations, both groups having benefit from it. A type of symbiosis in which one population harms another but remains unaffected itself is called amensalism. So in the case of bacteria, some amensalist species produce bactericidal substances that kill another species of bacteria. So in this example, one population is hurting the other population. So one population is harmed and the other population is unaffected. So for example, Mucilia sericata produces a protein that destroys Staphylococcus aureus, a bacterium commonly found on the surface of the human skin. So too much hand washing can affect this relationship, can affect the balance between the two, and lead to Staph aureus diseases and transmission. So we see where one is going to produce this protein that destroys the other. And then the first one, Mucilia sericata, for example, is unaffected by that. In another type of symbiosis called commensalism, one organism benefits and the other is unaffected. So up here, it was one population was harmed and the other was unaffected. Down here, one organism benefits, the other is unaffected. So this occurs when Staphylococcus epidermidis uses the dead cells of the human skin as nutrients. So in that case, the Staphylococcus is going to benefit, it's getting food, and we as humans are unaffected. It doesn't harm us, it doesn't help us. There's just this bacteria living on our skin that is able to eat our dead skin. So billions of these bacteria live on our skin, but in most cases, we don't react to them in any way. If neither of the symbiotic organisms is affected in any way, this is called neutralism. So neutral, it doesn't help or harm either. An example of neutralism is the coexistence of metabolically active bacteria and endospores. So we've already spoken about endospores. They are dormant, metabolically passive bacteria, right? Remember, they're not interacting with their environment, they're not metabolizing anything, they're not producing any products. So they, as endospores, can just kind of live amongst vegetative bacteria, and they're not going to be harmed. So Bacillus anthracis typically forms endospores in soil when the conditions are unfavorable. If the soil is warmed and enriched with nutrients, some endospores will germinate and remain in symbiosis with the other endospores that have not germinated yet. So they can all live together and it doesn't hurt one or help one. So another type of symbiosis where one organism benefits and the other one is harmed is called parasitism. This is the relationship between humans and many pathogenic prokaryotes. That's a parasitic relationship because these organisms are invading the body. It's good for them, they're benefiting, but it produces toxic substances or an infectious disease that's causing harm to the human. So diseases such as tetanus, diphtheria, pertussis, tuberculosis, and leprosy all arise from a bacteria-human relationship where, of course, the bacteria is getting something from it. It's able to metabolize body cells and utilize body cells in order to function, but then that's harming the human at that time. So scientists have coined the term microbiome to refer to all prokaryotic and eukaryotic microorganisms that are associated with a certain organism. Actually, before we jump into that, I just want to go through and mention these one more time. So keep in mind that we have all of these different interactions. So parasitism, where one is benefited and the other one is harmed. Mutualism, where we're not harming or benefiting either. Commensalism, where one organism is going to benefit, the other one is not affected. Amensalism, where one is harmed and the other one is unaffected. And then up here, mutualism, where both are benefited. Okay, so make sure you understand those and have some examples of those. And then this is the chart right here that summarizes all of that, table 4.1. All right, so then let's move on to our microbiome. So they have coined the term microbiome, and that refers to all of the prokaryotic and eukaryotic microorganisms within a certain organism. So within the human microbiome, for example, there are resident microbiota and transient microbiota. So the resident microbiota, biota or biota, whichever, resident microbiota consists of microorganisms that constantly live in or on our bodies. So in this example, this microbiome, we're talking about the human microbiome. So this is going to include all of the microorganisms that live on the body. And then those can be separated into two different groups. There can be resident microbiota and transient microbiota. So the resident ones are ones that are constantly living in or on our bodies. So these would be the kind that make vitamin K for us. These would be the kind that break down the cellulose and make it into usable parts for us. Then the term transient microbiota refers to microorganisms that are only temporarily found in the human body. So these could include pathogenic organisms. So they would be a part of our microbiome, but only for that period of time when we have that infection, for example. And as a note, hygiene and diet can alter both the resident and the transient microbiota. So if we change the way we eat, for example, change our diet for a long period of time, we can change the type of microbiome we have, the different types of microorganisms that we have in our body. And then that can, of course, bring in some sort of pathogenic bacteria, for example, that can give us an infection, and then that will go away, and that's a transient. The resident microbiota is amazingly diverse, so not only in terms of the variety of species, but also in the preference of different microorganisms for different areas of the human body. So for example, in the human mouth, there are thousands of commensal or mutualistic species of bacteria. Some of these prefer to inhabit different surfaces, so the tongue, the internal surface of the cheek, others the front or the back teeth or the gums. And we see something like the inner surface of the cheek has the least diverse microbiota because of its exposure to oxygen. So we see different types of bacteria in different areas just within the mouth. And then by contrast, the crypts of the tongue, so like the indents in the tongue, and the spaces between the teeth are two sites that have limited oxygen exposure. So these sites have more diverse microbiota, including bacteria living in the absence of oxygen. So we see different types of diversity. So we have less diversity on the insides of our cheeks, more diversity in between our teeth and on the tongue. And then we also see differences in the oral microbiota between randomly chosen human individuals. So from one human's mouth to another human's mouth, one human's inner cheek to the other human's inner cheek. So there are so many differences. It's amazingly diverse is the point here. So studies have shown, for example, that the prevalence of such bacteria as Streptococcus, Haemophilus, Neisseria, and others were dramatically different when compared between individuals. There are also significant differences between the microbiota of different sites of the same human body. So the inner surface of the cheek has a predominance of Streptococcus, whereas in the throat, the tonsil, saliva, there are 2 to 3 times fewer Streptococcus. So in the different areas, we see less of the same bacteria. So not only do we see differences in how diverse bacteria are, we also can see sometimes the same bacteria throughout the body, but in very different amounts, and several times more Fusobacterium. In the plaque removed from the gums, the predominant bacteria belong to the genus Fusobacterium. However, in the intestine, both Streptococcus and Fusobacterium totally disappear, and then we see Bacteroides. So just throughout that organism, we see things in very different amounts in very different areas, and we see lots of different types of diversity, less diversity, more diversity. Also, not only can the microbiota vary from one body site to another, the microbiome can also change over time within the same individual. So we're looking at life changes. So the microbiota of a person, even if they don't really change their diet, for example, they generally have kind of a similar diet. When they were a child, they will have a very different microbiome than when they are an older adult. Humans acquire their first inoculations of normal flora during natural birth and shortly after birth. So this is when we start to inoculate the body, basically, starting with the digestive tract. So before birth, there is a rapid increase in the population of lactobacillus in the vagina, and this population serves as the first colonization of microbiota during natural birth. So as the baby is being born, since there's this increase of lactobacillus in the vagina, as the baby's going through there, they're going to get their face and their mouth and their body covered in this lactobacillus. And then this is going to be the first colonization, the first type of bacteria that a newly born baby is going to interact with. And that's going to start the process of creating their microbiome. After birth, then, additional microbes are acquired from healthcare providers, parents, other relatives, and individuals who come in contact with the baby. So right after birth, then every time someone is picking up the baby or is breathing around the baby in the same room, whoever is there, they are shedding bacteria all of the time. And so whenever the baby is coming in contact with a new type of bacteria, that's going to start to establish their microbiome. And then that will continue to evolve over the course of the individual's life. So as new microbes colonize and are eliminated from the body. So for example, as estimated within a 9-hour period, the microbiota of the small intestine can change so that half of the microbial inhabitants will be different. So that is a very quick change. Within a day, you can change up to 50% of all of the microorganisms that are living in your intestines. The importance of the initial lactobacillus colonization during vaginal childbirth is highlighted by studies demonstrating a higher incidence of disease in individuals born by cesarean section compared to those that are born vaginally. So scientists are really looking closely at whether a child that is born by cesarean section, which doesn't have that opportunity to go through the vagina and pick up that lactobacillus, whether or not they have the same type of microbiome, which of course they don't. It's very different. And then what has been noticed recently is that those that are born via cesarean section tend to have higher incidences of disease than those that were born vaginally. And so they're looking at babies that are born vaginally, how their microbiome is different than those that were born by cesarean section. So studies have shown that babies born vaginally are predominantly colonized by vaginal lactobacillus, whereas babies that are born by cesarean section are more frequently colonized by microbes of the normal skin microbiota. So because they did not go through the birth canal, they were taken out of the uterus, and then their first interactions with bacteria would be those that were being held, so skin bacteria, including common hospital-acquired pathogens. So a lot of pathogens are at hospitals, and so if a baby is born via cesarean section at a hospital, some of those first interactions may actually be pathogenic ones, and then immediately cause decreased ability to fight off disease. Throughout the body, resident microbiotas are important for human health because they occupy niches that might be otherwise taken up by pathogenic microorganisms. So not only establishing one's microbiome, whether being born cesarean or vaginally, also we have these niches that are filled by certain organisms that will fight off pathogenic microorganisms. So for example, the lactobacillus species are the dominant bacterial species of the normal vaginal microbiota for most women. So we see that increase, of course, in childbirth, but that is a normal species that lives within the vagina for most women. And then lactobacillus produce lactic acid, which contributes to the acidity of the area and inhibits the growth of pathogenic yeasts. So having a high amount of lactobacillus is going to produce a high amount of acid, which is going to maintain that acidic pH. However, when the population of that resident microbiota is decreased for some reason, so for example, if one is taking antibiotics, if it's a general antibiotic, a broad spectrum, it can kill all of the bacteria that it interacts with in the body, including these lactobacillus that are found in the vaginal area. So then the pH of the vagina is going to increase, so it will not be as acidic, it'll become more alkaline, and then it makes a more favorable environment for the growth of yeasts, such as Candida albicans, and then that would be a yeast infection. So we have this very delicate balance between having a certain amount of bacteria living in a particular area, and then if that bacteria is affected in some way, if it's changed, if it's adjusted, for example, by taking antibiotics, then something that is more harmful can come in and take over. Antibiotic therapy can also disrupt the microbiota of the intestinal tract and the respiratory tract, increasing the risk for secondary infections or promoting long-term carriage and shedding of pathogens. So taking antibiotics is a very serious thing because it can wipe out the bacteria not only in the vagina for females, but also in the intestinal tract and the respiratory tract. And then that allows then the human, it makes them vulnerable. It makes a vulnerability where then pathogenic bacteria can come in or yeasts can come in and take over and then can cause more infections and cause more systemic issues. All right, so now what we're going to do is kind of switch over to taxonomy here in systematics and talk about Bergey's Manual of Systematic Bacteriology, just briefly again. We have talked about this already in previous chapters, but we're going to brush up on it for a moment and then move into actual bacteria, looking at certain classifications and different groups of bacteria. So assigning prokaryotes to a certain species is challenging. They do not reproduce sexually, so it is not possible to classify them according to the presence or absence of interbreeding. So it's very difficult to classify different prokaryotes because we're not able to look at its line, basically. They don't interbreed, so we're not able to say, okay, this is where this branched off, this is where this branched off very easily. Also, they do not have many morphological features, so they look very similar. Traditionally, the classification of prokaryotes was based on their shape, staining patterns, and biochemical or physiological differences. However, more recently, as our technology has gotten better, the nucleotide sequences and genes have become an important criterion of more microbial classification. So as we're getting more and more technologically advanced, now we're able to look at the DNA of all these different microorganisms and then classify them based on that. In 1923, David Hendricks Bergey published a manual in determinative bacteriology, and as we already spoke about in this manual, he tried to summarize the information known about different bacteria at the time, and then he used this Latin binomial classification, which we've discussed, the genus species. He also included morphological, physiological, and biochemical properties of these organisms. His manual has been updated multiple times. A more recent publication is this 5-volume Bergey's Manual of Systematic Bacteriology, and it expands on his original manual, so it's been kept up with. It includes a large number of additional species, up-to-date descriptions of the taxonomy, and biological properties of all these prokaryotic taxa. It also incorporates the approved names of bacteria as determined by this list of prokaryotic names with standing in nomenclature, the LPSN. This right here, this link, I just want to point out, can link you to Bergey's Manual of Determinative Bacteriology, which I think is interesting. So you can just click on this link here on your device and then you can access that. All right, so classification by staining patterns. According to their staining patterns, which depend on the properties of the cell walls, bacteria have traditionally been classified, as we know, into gram-positive, gram-negative, and then atypical, meaning neither gram-positive nor gram-negative. So we talked about that in the staining microscopic specimens in the previous chapters. Gram-positive bacteria have that thick peptidoglycan cell wall that retains that crystal violet stain. Then they remain purple. And then the secondary counterstain, safranin, is added. So then the gram-negative bacteria possess that thin peptidoglycan cell wall that does not prevent the crystal violet from washing away when we do the decolorizing step. So then we see that light red or pink after staining with the safranin. Bacteria that cannot be stained by the standard gram stain procedure are called atypical bacteria. So if this gram staining procedure does not work for them, it's atypical bacteria. So we see that in something like the mycoplasma and chlamydia groups. And this is because they lack a cell wall, so they can't retain the gram stain reagents. They can't retain the crystal violet or the safranin because they don't have that cell wall with the peptidoglycan layer in order to retain that. Rickettsia are also considered atypical because they are too small to be evaluated by the gram stain. So they're very, very tiny, and so they don't really pick up on the gram stain to where we can see that. More recently, scientists have begun to further classify gram-negative and gram-positive bacteria. So they have added a special group of deeply branching bacteria based on a combination of physiological, biochemical, and genetic features. They also further classify gram-negative bacteria into proteobacteria, cytophaga flavobacterium Bacteroides, which is the CFB group, and spirochetes. So we're going to look at each of these different groups also in just a moment. So the deeply branching bacteria are thought to be a very early evolutionary form of bacteria, and we're going to take a closer look at the deeply branching bacteria at the end of this chapter, but in general they live in hot, acidic, ultraviolet light exposed, and anaerobic conditions. So anaerobic, an is without, and then aerobic is air, so without air. So deprived of oxygen, without oxygen. Proteobacteria is a phylum of very diverse groups of gram-negative bacteria. It includes some important human pathogens, and we're going to talk about that in more detail. And then we have the CFB group of bacteria that includes components of the normal human gut microbiota, like we've talked about already, Bacteroides. The spirochetes, the last of the groups, are spiral-shaped bacteria, which include things like Treponema pallidum, which causes syphilis. And then we are going to look at these groups in more detail. So this was kind of the general overview, mentioning that these have been further classified into these different groups, and then we'll look at those different groups. We also have a little bit of a general group difference here with the gram-positive bacteria. So the gram-positive bacteria are classified into low GNC and high G plus C gram-positive bacteria. So let me back up just a minute. So up here, this section here, there's this special group of deeply branching bacteria, and then we also have gram-negative bacteria split up into the proteobacteria, the CFB group, and then spirochetes. So we're talking about, we've got deeply branching bacteria, which is a group, then we've split up gram-negative into these groups, proteobacteria, the CFB, and the spirochetes. And then what we're doing is coming down here, we're now splitting up our gram-positive bacteria into our low C plus G and our high C plus G gram-positive bacteria. So the low C plus G bacteria have less than 50% guanine and cytosine nucleotides in their DNA. So the G stands for the guanine, and the C stands for cytosine. And then remember, back to studying DNA, those are the nucleotides that are in DNA, the C, the G, the A, the T. We're talking about just the C and the G here. They include human pathogens, so these low C and G gram-positive bacteria include human pathogens such as anthrax, Bacillus anthracis, Anthracis, Clostridium tetani, and listeriosis, Listeria monocytogenes. High GNC gram-positive bacteria have more than 50% guanine and cytosine. So that's how we designate low versus high. Low C and G, less than 50% guanine, cytosine. High G and C, more than 50% guanine and cytosine. This includes bacteria that cause diphtheria. I say Corynebacterium, but it's Corynebacterium, diphtheria. I'm trying to say that better. Corynebacterium, diphtheria, tuberculosis, which is Mycobacterium tuberculosis, and some other diseases. And we'll look at that more specifically also. So this is generally how things are classified. We've broken up the gram-positive bacteria now into low GNC and high GNC, and these are some examples. So what we're doing in this chapter, in previous chapters, we've split it up by, when we've looked at bacteria, we split it up by gram-positive, gram-negative, and we talked about the differences between gram-positive cell walls and gram-negative and what that means. Now, in this chapter, what we're doing is splitting up our gram-negative groups and talking about those different types of groups and what that includes, and taking our gram-positive group and splitting that up into different groups, smaller groups. So we also will then look at other groups as well. Remember, there's the atypical group that doesn't fit into either of those, and we'll look at that, and then the deeply branching bacteria, which we'll look at also.